So thank you again to the organizers for the chance to speak to you today. Let's see if we can get back to number one here. Perfect. So I'm here representing a number of us who are interested in something we've sort of termed synthetic regulatory genomics. And where this stems from is really, you look at the slide, it should probably be titled Mammalian Gene Regulation. And what we're trying to represent here is really, as was alluded to in several talks before, the complexity of the regulation of genes, which gives us different cell types and fundamentally all of the interesting biology and medicine that we hear about. And what's going on this slide is this is your DNA fiber as it's packaged in the nucleus, wrapped around chromatin fibers. And you can see here that in these little pockets right here, where you have a bunch of transcription factors binding, in these regulatory DNA sites, this is where all the action happens. And the binding of these transcription factors is specified by sequence, but this sequence plays out in an epigenetic context that can have a lot of differences in terms of what effects you see in one cell type or another. And the point of having all of this up here is to give you a sense also that genomic context is important. If you move these elements, these regulatory elements around, and people have done this in fairly small scale in the past, they don't behave the same way in different regions of the genome. And so this is both a complication in terms of trying to study gene regulation as well as an opportunity for this group here. And classically, these elements were mapped originally by their accessibility to nucleases like DNase-1. These days, we can do this on a genome-wide scale, turn it into sequencing reads, and you see a lot of tracks like this, where each of these peaks represents a particular regulatory element. And so underlying these is going to be particular sequences that recruit particular transcription factors. And if you go ahead and look in different cell types, you can see that there's a great variety of the regulatory landscape in different cell types. So this has these elements are things that you may have heard of, like promoters and enhancers. These different categories, sort of regulatory elements that we try to assign. And they've been mapped in great depth over the last 5 or 10 years by very large national and international consortia like ENCODE, the Roadmap Epigenomics Project. And you can see here a list of a huge variety of cell and tissue types for which data is available publicly on the internet, identifying regulatory DNA in different cell and tissue types. And just to give you a sense of what this represents, in an individual cell type, maybe 1% of the genome is represented by these regulatory elements, but because they are so cell type specific, there's maybe upwards of 4 million distinct regulatory elements in the genomes if you take the union across all different cell types. So there's a lot of material out there. Now, the big question now is what do these elements do? And we've used these terms, promoters, enhancers, and some others that were assigned in the classical era using particular functional approaches, but it really doesn't scale to what we see today in terms of the genomics view of things. And this is just to give you a sense that you can look at the binding of multiple different transcription factors, coactivators, co-repressors. You can look at 3D structure, and there's a lot of things that are going on at a simple little locus here. So the question you might ask is, well, why hasn't this all been answered yet? And so I kind of wanted to, or actually, so step back just before we get there. As Neville and George mentioned, one of the reasons why this is such a pressing issue is that the bulk of energy in human genetics in the last more than 10 years now has gone into genome-wide association studies and is moving towards whole genome sequencing. And for the common diseases and traits that many of us are interested in, you can see here this is just sort of a summary of many studies that have been done over 1,000. The majority of the hits lie in non-coding DNA. So basically only 5% of those hits are landing in encoding regions where we think we could interpret their effect in terms of protein structure. The rest are landing in non-coding DNA, and they're highly concentrated in these regulatory elements. So this makes the function issue a very pressing one for personalized medicine. Okay. So to give you a sense of why has this been a hard problem to tackle, well, there's really two ways, essentially, or a spectrum of ways that between two different poles here of studying regulatory variation in its endogenous context, both cellular context and genomic context. And the first is engineering variation, and this has been going on for many decades, and has recently been accelerated using nucleases to create double-stranded breaks. And this gives you a pretty high degree of control at single site. And as you heard from Neville's talk, you can extend this to many sites with some technical complications. But there are some limitations in terms of what you are able to do. And as you want to start doing multiple changes, things get harder. The other approach has essentially been looking at natural variation. And so this is the idea that you can take polymorphism segregating in a population, and you can use genome-wide profiling techniques to identify functional variants in terms of some molecular trait, like gene expression or chromatin accessibility. And this gives you very efficient yield of functional variation, but you, of course, have no control over it because it's simply segregating polymorphism. We've done some work using chromatin QTLs, if you will, and essentially we've been able to build local models of the effect of sequence variation on TF activity. And so I think that there's been a certain amount of progress using this sort of endogenous approach for studying regulatory variation, but the real problems is that we don't essentially have reporter assays that are capable of studying regulatory variants with all the types of questions that we might like to ask. And these are just 3 axes here in which you can classify different reporter assays. And kind of the simplest thing to do is you can engineer yourself a plasmid, and you can clone in whatever a promoter or maybe enhancer next to it you want, and you can look at it in vitro or in transient transfection or so forth, and just do it on the scale of a single reporter. There are plenty of stories where the approaches from the results from these techniques don't map up with what is known when you start doing work at the endogenous locus in whole organisms. And so we want to be pushing this technology so that it's not so much reporter anymore. You start getting into much larger sizes capable of encompassing entire loci. You start going back in, integrating into the endogenous locus, and start doing this at much larger scale. So we are interested in pushing this forward, a group of us at NYU as well as at other places. This is just a quick overview of our strategy. You can see that we've developed, Jeff's lab has developed essentially a backbone vector into which we can integrate a large set of DNA, upwards of 100 kilobase pairs. It would contain a gene of interest, and at the places where there are these hypersensitive sites, we can replace it with modules that would let us put in whatever we want. And that is really taking advantage of all the work that's been done for mapping to focus our attention on the elements that are important so that we can really scale this problem and doing in-depth analyses of these guys. So the problem after this is really delivering these guys, and as I'd mentioned, we want to be doing this initially at the endogenous locus to replace one of the 2 alleles in a diploid genome. And so we've been extending past work to be able to do an efficient site-specific integration here. And then ultimately the interesting parts are going to be what you're going to do to profile these. And so one advantage of focusing on a particular locus is that there are quite efficient capture reagents similar to what's done for exome studies. And we can use that to go beyond just doing simple profiling of steady-state gene expression and look at local chromatin accessibility as well as chromatin conformation and their effect, the effect of various changes and substitutions that we make. And this opens up the analysis of a whole suite of things that really have been difficult to address in previous paradigms, especially making multiple changes, as well as this gives you the ability, once you've generated these DNAs, to bring them from one species to another, say mouse, human, whatever, without worrying about the existing sequence in the genome. Okay. There's a whole number of things that you can do here. You can make single substitutions, pairwise substitutions, and so forth. But our strategy is very, very simple. We scan through the first single set of all the single knockouts. We have the facility to do this rapidly enough that we're going to be able to go back and, based on those results, test double knockouts as well as testing other types of changes in sort of 3 waves per year. Let's see. Just to give you an example of what we've been working on, this is in collaboration with Doug Higgs' lab in the UK, and this is a plot from a recent paper of theirs at the alpha globin locus, showing you the 2 alpha globin genes in human as well as the locus control region. And essentially, they have done some very interesting deletions that they've brought into mice, and this has been very informative for the function of the locus control region around these genes. And essentially, we can go well beyond what they've done in the past by increasing the scale and starting to ask many new questions. Okay, so I'd mentioned this is a collaborative project, so my lab is involved at the same institute, Jeff Buca, his lab, as well as a team in his lab working on LIMS automation, which makes this efficient and possible, other collaborators, and funding for my own lab. Thank you very much for your attention. I'd be happy to take any questions. George.
So when you make heterozygotes, you don't have a problem analyzing the hypersensitive sites and so forth in complex heterozygous haplotypes?
So we've done a lot of work in the past, as I mentioned, looking at natural variation. So this is essentially the allelic imbalance problem that has been the basis of a lot of our work. So the advantage of doing this in this fashion is that you can actually put in point variants as markers. And some of the first waves of stuff are essentially going to be ectopic scrambled sequence, but we can go back in and put markers in for all these things, so we can perfectly distinguish the two alleles.
Perfect example of the advantages of synthetic over natural.
Absolutely. Thank you very much.